Hey guys, let's start.
Hello, and welcome to Data Science in Stratified Healthcare and Precision Medicine. I'm Frances Wong.
And I'm Arati Manataki. We are your course instructors, and we are delighted that you've decided to join us on this data science journey. In this course, we're going to explore how data science is transforming the life sciences, leading to precision medicine and stratified healthcare. We live in a digital world. We're generating data in our daily interactions with many things which we take for granted. This ranges from emails and social media posts to mobile phone records and bank card transactions. Data is all around us.
An increasing volume of data is also becoming available in biomedicine and healthcare, from genomic data to electronic patient records and data collected by wearable devices. Recent advances in data science allow us to integrate different types of data for every patient, to process it at a reasonably fast rate and to analyse it in ways that we couldn't even imagine some decades ago. Bringing different types of data together for each patient allows us to take a more personalised approach to therapies, tailoring them to suit each individual. This is precision medicine. Precision medicine is an emerging field and is transforming the medical approach to disease treatment and prevention. It focuses on identifying the most effective strategy for each patient based on genetic, environmental and lifestyle factors.
The ability to characterize individuals much more precisely then allows us to identify key differences across human populations and to act accordingly in healthcare provision. This is stratified healthcare. So one can consider stratified healthcare and precision medicine to go hand in hand. In this course, we will look at stratified healthcare and precision medicine from a data science point of view. There is no need for you to have a background in computer programming. This course is aimed at complete beginners in programming and computer science, and we're here to help you and support you in every step of this journey.
By the end of this course, you will have learnt about some of the different types of data and computational methods involved in stratified healthcare and precision medicine. You will have had hands-on experience of working with such data, and you will have learnt from leaders in the field about successful case studies.
Okay, let's move ahead.
So, how does the course work? It has a duration of 5 weeks. Throughout the course, we'll be looking at different data science topics in stratified healthcare and precision medicine. Each week's material will be delivered as short videos supported by readings. There will be optional quiz questions in the videos to help you verify that you've correctly understood new concepts. In order to help you reflect on the topics covered each week, we'll include additional videos of case studies with interviews of experts and professionals in the fields of data science and medical research.
The coursework includes weekly quizzes, a programming project in week 3, and an end-of-course assessment. There will be a quiz at the end of every week containing multiple choice questions, which will cover the specific topics you have learnt during that week. This will allow you to identify your strengths, and you may wish to revisit certain parts of the course. Apart from the quizzes, there will be a programming project in week 3, which will give you the opportunity to play with some examples of medical and healthcare data. In week 5, there will be an end-of-course assessment. This will be in the form of an essay, and you will be required to discuss a few of the topics that we have covered in the course. You will be able to demonstrate your
ऐसे
understanding
लिखना
and
पड़ेगा।
further explore your chosen topics. These essays will be peer graded, which means that they will be marked by your classmates and that you will mark the essays of some of your fellow students. We will provide you with a clear marking scheme so that you will feel comfortable with marking these essays.
One of the most important resources for this course is the discussion forum, where you can interact with your classmates to discuss the topics we cover every
Let's
week.
move ahead. So this is the syllabus. Okay. Introduction to programming in Python. So I think I already know this. Case study, data science in biomedicine and healthcare in Scotland. Okay. In sequence processing, medical image analysis, Edinburgh, genomic generation, Scotland, network modeling, probabilistic modeling, machine learning, natural language processing. Okay, sounds good, sounds good. IBM Watson's Arithmetic analytics platform. Got it, got it, got it. Mark this as completed. Why is what's not completed?
Classmates to discuss the topics we cover every week.
Okay.
Whenever you have a question.
So this is complete. This
For
is
case studies,
also complete. Right. I will move on to course logistics. Okay. That's fine. How to use discussion forums? Fine. Course team 10 minutes. Okay. Fine. Acknowledgements. Fine. Copyright. Yeah. I may as well just make this video private. It's just for me. Let's see.
In this course, we're going to explore how data science is transforming the life sciences, leading to precision medicine and stratified healthcare. But what is data science? Data science. This involves cleaning from the data, source and free to install.
Got it.
And value to the contains the
Basic Python. Not going to go through this. It's fine. How to use Jupyter? That's fine. All of this is very basic, very basic. So anyone who wants to watch this, they can. Okay, going to directly to the notebook. Let's see, it's a 1 hour. I guess I will complete it quickly. I hope I can. Okay. Let's see, you can run the code. Run away. Run away. Creating some variables. Okay, yeah, we have to do something here. Just okay. Is it okay? Oh, sorry. Okay, so it was a tuple and it failed. Fine. Yeah, it's given here. It should fail. Okay, so it's fine. What? Append dictionaries. Everything is fine. Data structure in pandas library. So they created a series, and they gave the index to the series, right? Right. Do I have to submit it? I don't know. I don't think so. And the save changes. Close. I don't think I do.
Please. Tabular data in plain text. But right now we don't know much about it. We can do all and for what part of our data? We'll keep learning about buying.
Oh, okay. I guess we are moving to week 2 also. So let's do that. Case studies, okay. Let's see what's in the case study.
My name's Andrew Morris. I'm a doctor. I'm the professor of medicine at the University of Edinburgh. I trained in medicine and diabetes care and also clinical pharmacology, which is the study of drugs in man. My major research interest is medical informatics. I'm vice principal of data science at the University of Edinburgh, which allows me to apply my research interests. I'm also director of the FAR Institute in Scotland, which is a major collaboration between 10 funders and the NHS in Scotland to bring together the collaborative strengths of informatics research across the nation. I think it was Bill Gates who said that healthcare is the last major industry not to be transformed by the information age. Indeed, if you look at other industry verticals, such as banking, such as retail, such as the airline industry, they have embraced data science to actually change their business model and actually improve the services that they provide to their consumers or the public. I think there's a huge opportunity to do this in healthcare. It's important, however, to define data science. What is it? My definition, it's the intersection where mathematics, statistics, computer science, in all its domains, comes together with healthcare and biomedicine. I think there's an immense opportunity at the University of Edinburgh to get that chemistry right, to apply the new findings, the new methods in computer science, mathematics, statistics to important questions and challenges in healthcare, and that's what we're doing. What we're seeing internationally is healthcare systems are challenged by the rise of chronic disease, increasing or even spiralling healthcare costs, and increased public expectation about the quality of care. So when we apply data science methods, it has to be to real clinical or population challenges. So can we apply data science to improve patient pathways, to make care and deliver care of higher quality, but at the same time as reduce cost, because we can't just go on adding to healthcare costs. So efficiency of care delivery is important, as well as safety, effectiveness, equitable healthcare, and patient-centered approaches to healthcare. There are many challenges, that's what makes it so exciting. One of the first challenges is the complexity of healthcare. We talk about the NHS in the UK, and we refer to it as if it's a single organisation. It's actually a confederation of different healthcare providers, whether it's the general practitioner, the pharmacy, the optometrist, or the hospital system. So using data science across patient pathways which touch upon these multiple healthcare providers is a challenge. The second challenge is that we need always to do this in a trustworthy way, and we need to be awarded trust by the public. So to be trustworthy, one needs to be honest, reliable, and competent. And it's very important as we develop solutions which share information, which can be identifiable, that we ensure that the privacy and the confidentiality and the governance is of the highest possible quality. The third issue is about capacity building. I think it's recognized internationally that the best performing health systems have people working within them who are data scientists. So they have skills which perhaps the UK has not invested in the past. So we're very keen to invest in the development of people's careers so they can be leaders, not only in the clinical domain or the managerial domain, but have computer science, mathematical and statistical skills to harness the tools of data science. In terms of healthcare in Scotland, we spend £12.5 billion a year delivering health and social care, which is a lot of money. That's for a population of just over 5. The benefit of Scotland is its size, in that we have a unified healthcare system, NHS Scotland, which delivers care for these 5.3 million people. And we have the ability to collaborate across the nation. So we work very closely with our university colleagues across the country in Aberdeen, Dundee, Edinburgh, Glasgow, St Andrews and Stratified. We've got very strong universities. But also we work very closely with our publics and also with NHS colleagues. So I think the size of Scotland is in its favour, and the ability to follow people across journeys of care across the nation. So in terms of our strategy, our research strategy, firstly, we must build on our strong scientific base. And we've got terrific universities in Scotland. Secondly, we're creating ecosystems which are data rich. So ecosystems that take forward care, research and education in a very integrated way. Thirdly, and it's the data science piece, we're trying to enable information and data science across Scotland so that we achieve knowledge through analysis of data. Fourthly, we're trying to reduce the regulatory burden to allow research to take place as efficiently as possible. Fifthly, we like collaborative arrangements, not only with other public sector bodies, but with industry, with the charitable sector, and with the public. And lastly, I think to be competitive internationally, we should position Scotland as a single research and innovation site when it makes sense to do so, so that we can have global impact. The FAR Institute is a major vehicle to deliver on data science in Scotland and across the UK. About 3.5 years ago, 10 funders came together, which was very innovative, to do something that was ambitious, bold and enterprising. They thought that medical informatics and data science in healthcare across the UK was a cottage industry. It was siloed. And the exam question was, could we gear this up to actually increase our impact, our capability and capacity on the international stage? So the FAR Institute currently represents a £39 million investment, and it's created 4 centres of excellence across the UK in health data science in Wales, a node in London, in the northwest focused on Manchester, and in Scotland. So I have the privilege to act as the director of the FAR Institute in Scotland, which is a real collaborative effort between the NHS in Scotland and 7 universities. In terms of our activity, our number 1 priority is to produce pioneering research using healthcare data, both structured data and unstructured data. Secondly, we're keen to capacity build, create the future leaders in health data science of tomorrow. Thirdly, we're very keen to work with our partners who are responsible for curation of data sets, so-called data controllers, to enable those data sets in a trustworthy way in trusted research environments. Fourthly, working with the public to really engage and encourage active public participation to shape our research programme. Fifthly, we're looking at the ethics, the governance, and the legal aspects of trustworthy data sharing. And lastly, we are encouraging partnerships between academic institutions with the NHS and in a trustworthy, transparent way with industry partners. And we think the UK has a great opportunity to be world leading if we can bring health data science with a very strong academic backbone and create alignment with health service delivery. It's such an exciting opportunity. I think there's lots of exciting things happening across the research landscape, One area is in so-called precision medicine. I think there's a realization that in future healthcare and medicine needs to be more predictive, preemptive, personalized and participatory. And I think that data science tools and methods will play a key role in improving our ability to predict and personalize diseases and healthcare and patient management. For example, the drugs we use about 1,000 drugs in clinical practice. If you look at how effective they are, they probably only have on average about a 50% chance of working in any specific individual. So if we're going to personalise treatment, we really need to know more about their clinical characteristics, so-called phenotyping, and link that with informatics, analytics and genomics. So I think the UK is in a prime position to see that convergence of informatics, analytics, genomics, with phenotyping to try and personalise therapy. Another example, from the other end of the spectrum, most healthcare information is actually unstructured. It's written in notes, it's in medical images. Can we use new computer science methodologies, whether it's artificial intelligence or machine learning or imaging informatics techniques to derive some medical in any specific individual? So if we're going to personalise treatment, we really need to know more about their clinical characteristics, so-called phenotyping, and link that with informatics, analytics and genomics. So I think the UK is in a prime position to see that convergence of informatics, analytics, genomics, with phenotyping to try and personalise therapy. Another example, from the other end of the spectrum, most healthcare information is actually unstructured. It's written in notes, it's in medical images. Can we use new computer science methodologies, whether it's artificial intelligence or machine learning or imaging informatics techniques to derive and codify phenotypes from unstructured data, or even create a longitudinal electronic health record populated by information derived from unstructured data? The opportunities are endless.
Okay. Professor Eileen Keel. The speed is fine.
Hello Eileen, thank you for joining us today. Please tell us a bit about yourself and your current role with the Innovative Healthcare Delivery Programme, IHDP.
Well, a bit of my background first of all. I'm seconded out of the Scottish Government to lead the IHDP. And in the Scottish Government for a long number of years I was Deputy Chief Medical Officer. And one of the things I did and still do is chair the Scottish Cancer Task Force. So I've been aware for a long time that there's loads and loads of cancer data out there, but it's not joined up, sitting in different data sets, different IT systems which don't speak to each other. So I had the ambition to try and develop a Scottish Cancer Intelligence Framework. And through a number of steps managed to get agreement to be seconded out to focus just on doing that. So what the IHDP is aiming to do is harness the power of informatics to deliver value to patients, healthcare professionals, and wider interests. We're doing that in collaboration with academia, with industry, and with the third sector. And we're tasked with focusing on cancer, first of all, with the caveat that we take the approach we're applying to cancer and apply it to other areas, clinical areas, in due course, demonstrating that we are a nationwide learning health system as we do that.
What is IHDP's vision?
Well, it's to join up all those disparate data sets that are sitting out there and use that joined up data, turn it into intelligence to improve cancer outcomes.
How can IHDP achieve this vision?
With difficultational. So I'll start with the technical solutions. We've got a very guddled landscape, if we can use that word, if we just made it up, in terms of NHS IT. We've never had a master plan. We didn't commission the same system for each clinical area. So we've got a legacy of very disparate systems that don't interoperate, at least not easily. And the idea of trying to interface those systems just to derive cancer data is very daunting. So my colleagues in National Services Scotland, which is a special health board, which amongst other things houses the Information and Statistics Division, ISD, as well as informatics expertise, had been for a number of years working on something called data virtualization. Now don't ask me too many questions about that because I'm not an informatician, I'm a clinician. But in essence, this is a software platform that sits on top of the data, wherever that data may be held, be it centrally in the national data sets or data sets at board level, for example, in Lothian, where we're sitting today. So the data can sit in those silos, and you don't have to move the data from one silo to another to join it up. The data virtualization is agnostic as far as where the data is, and you can visualize it and you can virtually link it when you're using that software. So when I heard about this, I thought, gosh, this could be the solution to join up any data. So I'm working very, very closely with NSS on this endeavour, and we've got a whole programme of work over the next couple of years with clearly delineated milestones so that we don't let progress slip. And a big element of that, applying that technology, will be building the Scottish Cancer Registry from its current position as holding a fairly limited number of data sets to an enhanced position with many more data sets, so it could act as the backbone for the Cancer Intelligence Framework. IHDP and NSS have worked out this joint proposal to turn the Cancer Registry into the Scottish Cancer Registry and Intelligence Service. So not just enhanced data sets being held within the registry, but many more analytical functions being applied to the data. So if a researcher comes along and says, well, I would like to link pathology with radiology with SMR6 cancer registry data, it will be much easier for the registry to do that because the data will be sitting in a cancer data repository or warehouse, whatever you want to call it, and linkable using using our unique patient identifier, the client number in Scotland, and using the data virtualization technology that I've just mentioned. So we're beginning to make a success of that. The registry has adopted additional data sets very recently in pathology, in outpatient appointments, and we're going to be adding, for example, very important in cancer now, molecular pathology and genomic data to that, in the next couple of years. So that's the technical solution. The information governance is a whole different ballgame. It's immensely complicated. Again, a very busy landscape around information governance. We have over 1,000 data controllers in Scotland. So tidying that landscape up is one ambition. Getting consistency of application of information governance guidance and rules is another challenge. Because at the moment, for example, the chief exec of every health board is the data controller. Now, the chief exec isn't going to be looking at every request for access to data, so that the decision making is delegated. And what we found as we went around the country a couple of years ago visiting the cancer centres was that there was real variation in how those rules were being applied, depending on who was being asked to consider the application. So lack of standardisation and therefore an element of subjectivity coming into these decisions. So I'm very keen to declutter the landscape and get a much more standardised approach to the imposition of information governance rules. We've got an opportunity in the digital health and care strategy, which government is putting together as we speak, to have guidance on the way forward with information governance there alongside whatever technical recommendations are going to be emerging. So an opportunity to achieve what I just outlined through that process.
You're also involved in the Cancer Innovation Challenge. Please tell us a bit about this and how this is bringing people together.
Well, there's £1 million sitting in this Cancer Innovation Challenge pot, and the Scottish Funding Council, who of course fund higher education in Scotland, have put that money up. The SFC have 6 innovation centres, and they were very keen to identify an area where some of them could work together, and they came up with the idea of cancer. In fact, Andrew was persuaded that it should be cancer. So we've got Data Lab, we've got DHI, Digital Healthcare Informatics, we've got Stratified Medicine Scotland, who are the most relevant in this area. And Data Lab are in the lead on this. So we've worked very closely with the innovation centres to shape the project and then to do a number of things. First of which was a data hackathon back in summer of 2016, which brought together, as you can imagine, a very widespread community of data scientists, clinicians, e-health people, and began the process of awareness raising of the challenge. And since then, we've launched 2 innovation challenges, separate streams, one around PROMs and PREMs, patient-related outcomes and patient-related experience measures, and the other around a very open data science challenge, more or less come and help us with any solutions you might have to improve data analytics around cancer data.
You've also participated in the Edinburgh Fringe Festival. Please tell us about your event and your experience of this.
Data sharing, as I've already hinted, in Scotland is not easy. And Steph had the idea that somebody should get out there to the Fringe, participating in the Cabaret of Dangerous Ideas, and raise awareness of the benefits of data sharing for cancer, and begin to get that public dialogue going a bit more. With here, we're building public trust. Now, we know that the vast majority of cancer patients strongly support the idea of their data being shared, not just for their benefit, but for the benefit of their families and the wider public. But not all of the public agree with that. Not all non-cancer patients agree with data sharing. So Steph had the idea that somebody should appear at this Cabaret of Dangerous Ideas, and I happened to be in the room when she raised it, and everybody else stared at the floor, and I thought, somebody has to volunteer, so I was it. And then David Robertson, who was then head of informatics, came on board, so the 2 of us did a double header on the subject of Dr. Data, the next cure for cancer. So it was way out of my comfort zone. But in the event, it went, I think, pretty well. So the format is you stand up, you lay out your pitch for 2 or 3 minutes, in the area of diabetes. HDP with taking the learning from cancer to other clinical areas.
Let's start the quiz then. Okay, what is precision medicine? Personalized approach to therapy stating members of each individual. The ability to characterize individuals much more precisely allows to identify key differences across human population and to act accordingly in healthcare provision. This is stratified healthcare. Okay. Yeah. What are the benefits of using data science in medicine and healthcare? It can help analyze patient records, strategies, first for particular medical condition. It can help identify the most effective treatments for a particular medical condition. Both of the above. Okay. Which of the following statements about the data science process is correct? The data science process is linear. No, it's iterative. Cleaning data is only really part of the data science is the final phase. Is the final phase of the data science process. No, data science process may be repeated several times throughout a project. That's correct. Complete the following code so as to get the type of P. Okay. Okay. Type of P. That's not. Which of the following code specifies a dictionary that maps patient to their doctors? That's funny because they just put in that Janet's doctor was. The following code specifies a data frame that captures information about patients. What is a code for getting the shape of patients? And what is the output? Okay, so output would be we have 4 columns. 3, 4, 5. Okay, 5 rows, 5 rows and 4 columns. So 5, 4. Mm-hmm. Mm. Interesting, interesting, interesting. 32, 50, 36. 42, 83. 48.6. What are the areas of convergence? So this was this phenotyping, informatics, analytics, and genomics. That's right. Which of the following statements below is incorrect? ISDP aims to harness the power of informatics to deliver value to patients' healthcare. Uh, okay, so yeah. Yeah, that's the one. Submit. Oh wow, I passed 90%. Which one was incorrect, dude? Oh, okay. Interesting. Whatever. So let's move on to the next one. Oh, I have to run this too. Okay. I guess I'll just run. Okay. I don't know what do they want. Oh, so okay, right. Oh, great. So now we are on week 2. Awesome. That's awesome. And I guess I'll just go in my courses. Right, yeah. So now we are at week 2. And the assessment. So 1 hour for image analysis programming task. Analyzing medical images. Some part in that. Okay. Let's start.
Precision medicine is about personalised disease treatment leading to targeted therapies for each individual. This approach takes into account the differences of each person, such as their genes, environment and lifestyle. Therefore, at the heart of precision medicine lies you as an individual. Every person is unique from every other person due to their genetic code. The genetic code is a sequence of DNA letters that can be arranged in many different ways. So everyone is unique due to their order or sequence of DNA letters being different from everyone else's. A person's entire genetic code is called its genome. Your genome is your unique sequence of DNA. How do we know the sequence of the human genome? We know this because of the Human Genome Project. The Human Genome Project was an international collaborative research project. It aimed to sequence and map all the genes in the human genome. It was officially completed in 2003, enabling the genetic makeup of a human being to be known. This would not have been possible without sequencing technology. Further advances in genomic sequencing is enabling fast and accurate analysis of our DNA. Putting our DNA results together with a better understanding of diseases such as cancer allows for the development of more effective treatments, ensuring that an individual receives the right medicine to treat their particular condition. In the next few videos, we are going to tell you more about DNA, how it is sequenced and how it is analysed.
Wow, this is interesting.
DNA. What does it look like and how is it sequenced? DNA, or deoxyribonucleic acid, is a large molecule that contains genetic material and can be found in all living organisms. Biological information is stored in DNA as a code made up of 4 chemical bases: adenine A, guanine G, cytosine C, and thymine T. The sequence of these 4 bases encodes the information available for building.
Okay. I guess I need to write this up. Deoxyribonucleic acid. This is made up of 4 things. One is adenine, next is guanine, next is cytosine, thymine. Okay.
And maintaining an organism. There are over 3 billion bases in human DNA and is found in almost every cell in the human body. DNA bases pair up with each other, specifically A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar and phosphate are called a nucleotide. Nucleotides are arranged in 2 long strands called a double helix. The structure of the double helix.
Oh my god,
is like
this
a
is
ladder
like
a spiral.
this is too much
Nucleotide,
information.
a sugar
Okay.
molecule and a phosphate molecule.
Okay. I think I need to slow it down.
Bases pair up with each other, specifically A with T
and C with G to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar and phosphate are called a nucleotide. Nucleotides are arranged in two long strands called a double helix. The structure of the double helix looks like a ladder of two long strands called a double helix.
So this is called nucleotides, and its structure is a double helix. Okay. No, okay, I'll just draw this out. Okay.
The structure of the double helix looks like a ladder twisted into a spiral. The base pairs form the ladder's rungs, and the sugar and phosphate molecules alternate to form the verticals, form the ladder's rungs, and
the base pair forms the ladder's rungs. Okay.
sugar and phosphate molecules alternate to form the vertical sides of the ladder.
So this is the base pair, and this is the sugar phosphate backbone. Got it? Hey, this is interesting, man. Nice.
Often referred to as the sugar phosphate backbone.
Okay.
The two strands of DNA run in opposite directions to each other and are thus anti-parallel. Therefore, the order of bases on one DNA strand or side of the ladder determines the bases on the other side of the ladder. DNA strand or side of the ladder determines the bases on the other side of the ladder. Hence, DNA sequences are usually written as if DNA were single-stranded. Scientists only need to sequence one DNA strand in order to know the sequence of both strands. Genome sequencing is the process of determining the precise order of nucleotides or bases in a genome. This means the order of A's, C's, G's and T's that make up an organism's DNA. It is the order of these nucleotides along a single strand
treated as a single strand because identify one, you can guess the other. And now we move on to the genome sequencing. So genome sequencing is nothing but the order of these nucleotides along a single strand. Make sense? Make sense?
and that forms the genetic code. Today, genome or DNA sequencing is done by sequencing machines.
Okay.
These machines are high-tech but cannot sequence the whole genome in one go. Instead, they sequence the DNA in short pieces around 150 letters long. There are different methods and machines that can sequence genomes. The first DNA sequencing technology, called Sanger sequencing, uses a technique known as capillary electrophoresis, which separates fragments of DNA by size and then sequences them by detecting the final fluorescent base on each fragment. This technology was a breakthrough and was used to sequence the DNA from the Human Genome Project. It is a highly accurate method, which is time-consuming and expensive. Nowadays, we have the option of a more rapid and lower-cost technology, known as next-generation sequencing or high-throughput sequencing. With the sequencing technology, many strands of DNA can be sequenced in parallel, which significantly reduces the time from experiment to data. However, this generates more data per instrument run, meaning it will require larger data storage and the ability to analyze and manipulate larger data sets. Automated DNA sequencing machines output data in the form of peak trace chromatograms. This is a 4-color representation of the DNA sample. More specifically, it is a graph displaying the signal intensities of each nucleotide. Chromatogram files are binary files that not only contain the string of DNA bases, but also contain quality value information, such as confidence scores for the base call. Specific software tools are needed in order to open chromatogram files. Chromatogram files can be easily converted to sequence files, which are basically text files that contain strings of DNA bases. These files can be opened with a simple text editor. However, it should be noted that these files are in a rudimentary format and do not contain all the information you may need, such as confidence scores for the base call. In order to obtain good sequencing results, it is best to examine your chromatogram file rather than just using the text data from the sequence file. However, once you have good sequencing results for your chromatogram file, you can convert it to a sequence file and use this for further analysis. Each short DNA sequence from a sequencing machine is called a read. Chromatograms can have high quality, medium quality, and poor quality reads. High quality reads show no ambiguities, no noise,
Okay, guys, let's start again.
Multi-value information, such as confidence scores for the base call. Specific software tools are needed in order to open chromatogram files. Chromatogram files can be easily converted to sequence files, which are basically text files that contain strings of DNA bases. These files can be opened with a simple text editor. However, it should be noted that these files are in a rudimentary format and do not contain all the information you may need, such as confidence scores for the base call. In order to obtain good sequencing results, it is best to examine your chromatogram file rather than just using the text data from the sequence file. However, once you have good sequencing results from your chromatogram file, you can convert it to a sequence file and use this for further analysis. Each short DNA sequence from a sequencing machine is called a read. Chromatograms can have high quality, medium quality and poor quality reads. High quality reads show no ambiguities, no noise, and the peaks are very well spaced. This is the ideal chromatogram where the base calls can be trusted. Medium quality reads shows some ambiguities, but the peaks are still well spaced. However, there are some long single nucleotide stretches that are not well resolved. Poor quality reads show some ambiguities with a very small difference between the noise and the signal. The overall amplitude of the signal is small, with overlapping peaks and low confidence in base calls, giving a low confidence score. In general, predictable errors occur near the beginning and at the end of any sequencing run. However, other errors can occur in the middle. Therefore, it is good practice to check your chromatogram by eye to ensure your sequencing result is correct and to identify any potential errors made by the sequencing software. Sometimes the software will miscall a nucleotide. This means the peak was ambiguous and should have been called as N. Or occasionally, the software will call an N when the human eye would be confident in making a specific base call. Another example could be if the noise level is a bit too much, then you can check the signal intensity numbers for your chromatogram. In summary, a good chromatogram should show high peaks that are evenly spaced, each with only one color, and a low noise baseline level so that the real peaks are easy to distinguish.
Interesting. Let's go to the conclusion.
DNA R6.
This one is sequence processing.
In bioinformatics, sequence analysis is the processing of DNA, RNA, or peptide sequences by a range of analytical methods to understand its structure or function. Methodologies used include sequence assembly, sequence alignment, and searches against biological databases. Sequence assembly means the reconstruction of a DNA sequence by aligning emerging small DNA fragments. These small DNA fragments are the results from the sequencing machine. Thus, the sequence assembly process involves comparing sequences, finding overlapping fragment pairs, merging as many fragment pairs as possible, and creating a consensus sequence from the merged fragments. The challenges in sequence assembly programs are to allow potential sequence ambiguities and yet discriminate between
repetitive regions, gene family members, or genes sharing the same motif. Sequence alignment is the method of comparing two or more sequences by looking for a series of individual characters found in the same order in the sequences. There are two main types of sequence alignment: pairwise sequence alignment and multiple sequence alignment.
Okay.
Pairwise sequence alignment only compares two sequences at a time, whereas multiple sequence alignment compares many sequences. In order to find similar regions of a sequence, it is common to perform a search using a sequence analysis tool. A popular and widely used tool is a program called BLAST, Basic Local Alignment Search Tool. BLAST is a pairwise alignment tool and used to search for sequence similarity. More specifically, it is an algorithm for comparing primary biological sequence information, such as the nucleotides of DNA sequences. A BLAST search enables a researcher to compare a query sequence with a library or database of sequences, and identify library sequences that resemble the query sequence above a certain threshold. These analytical methods can aid precision medicine, for example, by providing a better understanding of genes involved in rare diseases. This in turn can lead to the development of targeted therapies. To put sequence analysis in a medical context, sequencing results from a human DNA sample can be mapped to a widely used reference sequence. This reference sequence was produced from the Human Genome Project. The reference sequence shows the position of our genes, most of which are known. Sequence analysis identifies the differences, called variants, between your genome and the reference. Most differences are harmless. However, some differences could be causing a particular medical condition. Genetic testing can be used to find changes that are associated with inherited disorders. The results of a genetic test can confirm or rule out a suspected genetic condition, or help determine a person's chance of developing or passing on a genetic disorder. For example, genetic testing can be done for cystic fibrosis. Cystic fibrosis is a genetic condition which usually affects people from birth and causes a range of symptoms. The main symptoms affect the lungs and digestive system. A genetic test for cystic fibrosis will look for the most common changes in the cystic fibrosis gene. If you have a family history of cystic fibrosis, you can be tested to determine whether you are a carrier of the defective gene that causes this condition. Sequencing data is an important part of precision medicine, as its analysis enables personalized treatment and prevention of disease. Each individual will benefit from targeted therapies, meaning that fewer people will suffer from unnecessary side effects that may occur with ineffective therapies. Precision medicine aims to provide better prevention and treatment methods for each individual, none of which would be possible without the individual sequencing results. Role with Edinburgh Genomics.
My name is Tim Aitman. I'm Professor of Molecular Pathology and Genetics in the University of Edinburgh, and for the last 20 years or so I've been researching into the genetics of rare and common diseases and particularly in the last 10 years, have been using information and technology about the genome to give insights in those fields. When I came to Edinburgh 3.5 years ago, I was asked to set up a genome sequencing facility at Edinburgh Genomics, and as a result of that, I'm the clinical director of the whole genome sequencing facility at Edinburgh Genomics. Edinburgh Genomics is based on 2 campuses, one of them at King's Buildings, and that has a wide range of sequencing technologies, animal genomes, and took 50 very high throughput activity. And this is where the skill is derived, because we then have to decide how significant that is for the protein function. And of course, a human genome is a lot of information. It's 3,000 million letters, or base pairs as we call them, and those are usually covered 30 to 60 times when one sequences the genome. And this is why we have a requirement for analyzing and storing terabytes of data. And in fact, the computational side is almost as important as the sequencing technology. And we have a compute facility where we regularly store about 2 petabytes of data, and we can expand that fairly easily to 5 petabytes. This is quite a substantial computing facility, and is one of the key reasons why the facility has been successful.
How many genomes are sequenced per week at Edinburgh Genomics?
So in terms of the throughput of the facility, we have a capacity for sequencing several hundred genomes per week. We usually are sequencing around 150 to 200 genomes per week. And I should explain how that can happen, because as you can imagine, sequencing a genome is not a trivial or straightforward task. Particularly, it's remarkable when you think that we only have 3 technicians or research assistants in the labs who carry out the lab work. And the reason that they can do that is because, firstly, we have a remarkably efficient laboratory information management system. Quite costly, but it means that we track the samples through from the very beginning to the end. And each time a new procedure or a new person is involved with handling that sample, it talks to the laboratory information management system and tells the system where the sample is. And in addition to that, every sample is handled almost exclusively by robots. So the robots obviously are programmed by humans, but because it's robotic, it can be very high throughput. And so we can easily handle 8, 16, or up to 48 samples at a time, meaning that we can have this very high throughput with very few people in the lab.
What are the main challenges in genome sequencing and analysing sequence data?
What are the main challenges in analysing human sequence data? Well, part of this relates to the volume of the data itself, because we cover each part of the genome 30 times, and there are, as mentioned, 3 billion base pairs in the genome. And so each of those segments is sequenced in small pieces, and each of those small pieces goes into individual computer files. So we have millions of computer files as the immediate output of the sequencer. Those have to then be joined up, and we join them up by mapping them to a reference genome. And that is done by now fairly standard software, and those reads are mapped to the genome, piled up, and then compared to the reference genome. When we find differences between that individual's genome and the reference genome, we call that a sequence variant. And there are around 4 million variants in any one individual's genome compared to the reference. And then we can say, are any of those significant? How do we assess their significance? We look to see whether they're common in populations, which part of the genome they're in. For example, are they in a gene that codes for a protein, or are they in regulatory DNA between genes? And if they're in a gene itself, what are the predicted effects on the protein structure and activity? And this is where the skill is derived, because we then have to decide how significant that is for the protein function. And for example, if we have an individual with a genetic disease or disorder, then we can say, could any one of those 4 million variants, or the smaller number that are within genes, for example, could they be responsible for that individual's disorder, and therefore could we make a definite genetic diagnosis from that person's genome sequence?
Wow.
How has genome sequencing helped in unravelling the genetics of both common and rare human disorders?
Well, genome technology has really revolutionized our ability to find the causes of genetic disorders. If we look at the first part of that, there are around 7,000 genes that cause human genetic disorders. And 20 or 30 years ago, we really knew almost none of those. Now we know about 4,000 of those, so probably over half. And most of those have been found because we've been able to sequence the genomes of people with a particular disorder, look for variants or mutations that are in common between individuals with that disorder, including individuals in the same family, and then say, do we know the cause? So that's the first part of it, that new genome technology has helped us to establish the cause of thousands of genetic disorders. And then the second part is, if we have an individual with a genetic disorder, can we track down the cause? And once again, genome technology really now is the key, to the extent that if an individual presents with a presumed genetic disorder, many people would believe, and increasingly it is becoming so, that the way to find out the cause of their disease is simply by sequencing their whole genome, analysing it for what variation they have in their genome, and saying, can we find what is often a single letter or single base pair change that causes that individual's disease. This is quite a remarkable change when one considers that the first genome sequence was only available just about 15 years ago.
What do you see for the future of genomics?
So the future of genomics, that's a fascinating question that geneticists, genomicists and social scientists all have a different perspective on. Just this week, I proposed a motion in the annual Edinburgh medical debate that the UK population should have genome screening. And what we agreed was that genome sequencing was almost certainly accepted by the audience and the speakers to be the method of choice for diagnosing genetic disorders. And so when one sequences a genome, the results are not completely certain. It may come up with a definitive diagnosis, but because there are 4 million variants, we often don't know what quite a number of them do. In fact, the overwhelming majority, we won't know what they do. And in addition, just because we're looking maybe for the cause of someone's genetic disorder, we will find other variants that might be responsible for other genetic disorders. And so if one's going to sequence someone's genome, even in the context of a disorder, one needs to have consented that individual and inform them about what the results are likely to be, both for the information about their own disorder, if they have one, and for any other information that comes about. And those things need to be very tightly regulated, and individuals need to be consented and informed about what the possible results might be and what implications they might have, for example, for their insurance and their future healthcare. So I think that in the context of genetic disorders, most people agree that genetic testing is usually beneficial. But then the question arises, if we can sequence a genome for under £1,000 and give people their results back, what about people who don't have an obvious genetic disorder? Maybe they have a family tendency to more common diseases such as diabetes or heart disease or psychiatric disease. Can we perhaps predict or give them some indication as to whether they are likely to be predisposed to these disorders? And the answer is yes, although we can't do it very precisely, and the information that we provide people with might give them a 1.5 or 2-fold increased risk, as opposed to the single gene disorders, where if they have the gene, they're almost certain to get the disorder, such as Angelina Jolie found with breast cancer and decided to take action by having mastectomies and ovarectomies. So there is information that healthy people may derive about their future healthcare, and the question is that beneficial? It's certainly technically possible. £1,000 and a few days of a sequencer and a scientist to analyse it, it's definitely possible. But it does raise the question, should we be sequencing babies at birth? Should we be sequencing children so that they know what the future is going to hold for them? And very strongly, I think, most people believe that we should not be sequencing genomes of people who cannot consent, which means that babies and children under the age of consent should not have their genomes sequenced. But maybe later in their life, there was a view, quite evenly balanced in the medical debate, that people will benefit from having their genomes sequenced. In the next 5 years, it's likely that worldwide between 50 and 500 million genomes will be sequenced. And so the question is, who should be doing it? Who should have their genome sequenced? Who should be regulating it, if any? Or should people just be able to choose to have their own genome sequenced? I think that there are risks of just allowing people to have their genome sequenced without any regulation or counselling. But I think there are also advantages. Although there will be costs to the healthcare system, in the long term, I believe it will make us a healthier nation and better informed about our futures and better able to make sure that if there is predisposition to disease, we have the opportunity for early and the best treatment.
Thank you very much for joining us today, Tim.
It's a pleasure.
PhD. My main interest is in using genetics to understand human conditions, and I'm a research professor here at the Institute of Genetics and Molecular Medicine.
Please tell us about Generation Scotland and why it is a valuable resource.
Well, Generation Scotland is a project that I started up when I was still in the MRC Human Genetics Unit, but it became very clear that it was a project that would actually benefit from moving to the University of Edinburgh, which I did in 1999. The idea behind it is twofold. First, that we could anticipate that the Human Genome Project would allow us to look at genetic factors that influence health in much, much more detail than we could ever have imagined before. And secondly, that we are at a time of considerable change socially and health.
Okay, be right back.